was less crucial because I know that I have another set. But this semester is last year. Well, digestion. Today we are going to talk about digestion in daily testing, and some mechanism of motor activity and hunger stress. In testing, our body has two principal parts of the intestine. The small intestine and large intestine with absolutely different functions. Small intestine can be divided to significant parts, duodenum and the rest. Because duodenum is the main process of the breaking down. Duodenum, there is digestion of all types of nutrients, all types, carbohydrates, proteins, and fats, by three juices. What juices? What juices act on food?
Pancreatic, bile, intestinal juices.
Intestinal juice. Or intestinal juice. Bile doesn't contain enzymes, but bile helps to activate some enzymes, for example, lipase, cholesterol esterase from pancreatic juice, and other functions, a lot of functions.
It doesn't have an enzyme.
No, it doesn't have.
I'm sorry, sir.
It's your problem because this.
It seems that, well, mine is
I will draw a big picture of the gastrointestinal tract. At first, talk about duodenum, and then other departments, other divisions, and some mechanisms. Oral cavity, esophagus, stomach, lower esophagus sphincter, esophagus sphincter, to control swelling, then stomach, next sphincter between stomach and duodenum, what is the name? Pylori. Pylori. Pyloric sphincter, then duodenum. Pancreas and bile, gallbladder have the common duct.
Upper and lower esophagus sphincter. What is that?
Lower esophagus sphincter, esophageal sphincter, adjective, and upper esophageal sphincter. Pyloric sphincter, then duodenum, pancreas. I will draw a simplified scheme of pancreas. Pancreas has exocrine part and endocrine part. Exocrine part releases juice. So now I draw a simple scheme of exocrine part, duct, and small ducts, ending with acinar cell, similar salivary glands, the same principle. Acinar pancreas cell and ductal cell or duct cell both produce pancreatic juice. Duct from gallbladder, bile, and what is a sphincter? Odi. Well, and the rest part of small intestine, then large intestine. Large. Ну, такая simple scheme of gastrointestinal tract. Pancreas. Да? Contains endocrine part. Endocrine part
Secretes hormones.
consists of islands. What islands?
Islets of
Islands.
Islets of
Lung cancer. Islets. Inside one islet, we can see different types of cell. Main cell, principal cell, alpha and beta. What hormone is released by alpha cells?
Insulin.
No.
Glucagon.
Glucagon into blood. And beta.
Insulin.
Again, into blood. So here we have T cell, inhibitory cell. T cell. The same in stomach. T cell produces somatostatin. Somatostatin. This cell exists in all parts of the intestine, mostly duodenum, and also in the stomach. These cells. I explained previous class, maybe no, I don't remember which group. This cell inhibits production of histamine or gastrin. Histamine. It's the same principle. Somatostatin inhibits production, release, to control release of acidic gastric juice, because excess of acid can damage glucose. It's dangerous. Each part of the most important parts of gastrointestinal tract, stomach and duodenum, have proper control, inhibitory control. Mostly with somatostatin, but not also somatostatin. Yes, release histamine affecting secretory cells in the fundus, G-gastrin. Gastrin affects growth blood. Gastrin and histamine stimulator hormones, somatostatin inhibitor hormones. Very about pancreas. Pancreatic juice compounds includes organic and inorganic. Organic part enzymes. What cell from here produce enzyme? What cell? Acinar or duct cell? Acinar cell. Here we have enzyme. Enzyme, exocytosis, enzymes. What is the function of duct cell? Bicarbonates. A lot of bicarbonates for what?
For neutralizing media in duodenum that HCl from
To neutralize hydrochloric acid and make alkaline pH. Alkaline pH. It's from stomach. Kind. From stomach, acidic kind. Acidic pH. Duodenum has alkaline pH. This pH must be alkaline, about 7.5, for example, alkaline. pH alkaline. Which is very important to understand the different function of acinar cell and duct cell. There is the same principle in saliva. Acinar cell produce both inorganic and organic, but duct cell transform inorganic composition. And the enzyme, you must know exactly enzymes of pancreatic juice. Enzymes of pancreatic juice. Some enzymes affect carbohydrates. Some enzymes affect proteins and lipids. Proteins and lipids. Well, now you. Folate. Right here, these enzymes are breaking down carbs and lipids. Right, by beautiful, right? Carbs. What enzyme breaks down carbs? What enzyme in pancreas? Pancreatic juice. What enzyme breaks down carbohydrates? I prepare to cross. One. But who can to read presentation before sleep is not enough. It's necessary to digest information. Carbs. What enzyme breaks down amylase, alpha amylase. Amylase, Y, amylase, alpha amylase, lipase. And what is a main enzyme? What is the main?
From pancreas, they are first in active form and active.
Yes, that's it. Proteolytic enzymes, proteases, are released in inactive form. What is the main?
We have trypsin, trypsin, and carboxypeptidase alpha and beta, A and B, and
Right here, everything active form. Active form, final form. The proteases are released as precursors, and then in the duodenum they transform into active form, converted into active form.
The enzyme in the
What an enzyme, enterokinase.
affects
Right
trypsin.
here. Enterokinase.
Affects trypsinogen and change convert it to trypsin. Trypsin is the active form and it's step by step activate.
Very good. Who are you? Forget Faisal. Pfizer.
Okay.
But you must know this exactly. To colloquium, for colloquium, the name of enzymes and their substrate. Alpha amylase active, active. Lipase, more or less. Lipase can work without hydrolysis of molecule, hydrolysis of enzyme molecule. But lipase requires additional activation, superficial activation by bile salts.
With emulsions, first emulsion of the
Bile cells emulsify fat, prepare to digestion, and help to increase activity lipase. All day, lipase has some activity. Trypsin, kind of trypsin here, CH. Trypsin is absolutely inactive. Chymotrypsin, carbopeptidase, elastase are both inactive. Both are for inactive. To escape self-digestion, to escape damage of secretory cell, acinar cell, because their proteolytic enzymes, proteases, they can break down proper components of our cells, pancreatic cells and tendons. Moreover, acinar cell produce special trypsin inhibitor, special substance helping to inhibit activation of trypsin until release of pancreatic juice into duodenum. Because sometimes with problem motor control, some portion of content, duodenal content, can pass inside pancreas. Trypsin is active here, it's dangerous, so it's necessary to escape. Enterokinase originated from the intestine, small intestine, the duodenum, and activates trypsin. Trypsin activates the rest. Pancreatic juice has another enzyme, cholesterol. Maybe you studied in biochemistry. Cholesterol esterase. It's more important for medical students, but for this I didn't put it in presentation. But I say it because maybe you studied in biochemistry to escape mismatch. Cholesterol esterase. It breaks down cholesterol to release free cholesterol esters, to release free cholesterol and free fatty acids. It requires also superficial conformation. What is the difference between activation lipase and trypsinogen? Trypsinogen enzyme is protein, of course, protein molecule, like each is enzyme, every enzyme is a protein molecule. Protein molecule has inhibiting part, and enterokinase breaks peptide bond to clever, separate inhibitory parts. And this enzyme becomes active. For example, enzyme molecules, here inhibitory part, and enterokinase breakdown, and the rest becomes active, this principle, to destroy peptide bond. And lipase and cholesterol esterase are not required this hydrolysis of peptide bond. But they have weak activity and they must change conformation with bile salts. It helps to increase activity. Well, regulation of pancreatic juice release. What regulation prevails in the duodenum? Humoral. Of course, numerous reflexes exist, but humoral is more important. If you cut, damage, destroy vagus nerve, volume of pancreatic juice reduces, but it will have. If you stimulate just vagus without hormones, pancreatic juice starts to release, but this effect is short-term, short effect, and then disappears. So vagus nerve has trigger activity. Vagus nerve, medulla, cephalic phase, preganglionic neuron, vagus nerve has postganglionic and stimulation. It stimulates pancreatic juice release, but this effect is short, it's prepared to digestion immediately after eating or during eating. If you measure increase of gastric juice volume and pancreatic volume during starting eating, gastric juice release starts immediately. Because when you start to chew, gastric juice is released immediately, without delay. Maybe very small. Pancreatic juice is released slowly. It's released approximately in 5-7 minutes after starting to eat. Because pancreatic juice requires humoral signal. Humoral regulation is slower. It requires time. Vagus has trigger effect. To increase volume of producing and releasing pancreatic juice, but it's not substantially compared with hormones. What are the main hormones? What are the main hormones working in duodenum? Cholecystokinin and
secretin.
secretin.
She said secretin and cholecystokinin.
Two hormones, cholecystokinin, cholecystokinin and secretin. What signal, continue, what signal stimulates the release of cholecystokinin? What signal from the duodenum?
The fat and protein break in that.
Fat and protein hydrolysis, peptides and fatty acids. The first product of the products of fat and protein hydrolysis, shortly fat proteins, because they have some destruction in the oral cavity, little proteins in stomach, Secretin, hydrochloric acid. There's a different stimulator. Stimulator for cholecystokinin cell, secretin cell. What is the main effect of cholecystokinin on pancreatic juice release? Acinar cell, well.
They produce, they release the enzyme.
Enzymes. There's more food, more cholecystokinin, more enzymes, especially for lipids and proteins. What is the effect of secretin?
Secretin works on toxins, they release big biochemical.
Well, thank you. There is the separate influence, prevailing influence. Actually, cholecystokinin and secretin can affect on everything here, but we talk about prevailing effect, main effect. There is some difference. Cholecystokinin act mostly at acinar cell, hydrochloric acid, or secretin, sorry, duct cells, from duct cells.
Professor, you write it in this here, محرک‌ها of cholecystokinin حضور فرآوردهای هضم غذا به خصوص مونوگلیسیرید. Just
Monoglyceride. For cholecystokinin. Yes, it's product of lipid digestion. Details. When I was listening to this lecture in Persian, I think this is something like this. And okay, this slide about
YouTube.
cholecystokinin. I don't know some details. Just understand about what. It's so much for me. I know more about the first topic, physiology, excitable tissue. But here it's not so good. Just compare with information, keywords. Yes, keywords. Cholecystokinin, shortly it exists in my presentation. Product hydrolysis of protein and lipids. Where? What happens with proteins in oral cavity? In oral cavity, what happens with proteins? Here, in mouth. Can proteins be digested in oral cavity? Very little, not substantial. But main digestion of proteins occurs in stomach.
And then intestinal, and then
Yes, the principles read. The oral cavity, some proteases mark protein molecules to inform brain about protein composition. Protein existence. Stomach breaks protein, denaturates proteins, and breaks to big molecules, big peptides. And here, peptides. Oligopeptides. Oligo means several amino acids, more than 3. 4, 5 amino acids inside or more. Oligopeptides. Big molecule break down to smaller parts. But this part, anyway, more or less big. Oligopeptides, proteins. Can stomach digest fat, lipids?
A little bit because in the oral cavity.
Oral cavity and gastric juice.
but the special organ is bile duct do this.
Yes, it doesn't contain bile, but gastric lipids doesn't require activation. It can digest fats. But fat molecule connect to big drop. So the surface of lipids is not available, for good digestion, effective digestion. So lipase, gastric lipase, chief cell in the stomach produces pepsin, pepsinogen, and gastric lipase. Gastric lipase is active, but substrate is not well available. So digestion of lipids here not is not effective. But after passing chyme into duodenum, chyme contains oligopeptides, some fatty acids, the products of lipid digestion. It activates cholecystokinin. Cholecystokinin stimulates and release more and more enzymes. But it's continuous process. Before digestion, during fasting period, every time gastrointestinal tract releases juices, every time. Even between meals during fasting period. More in the daytime, less in the nighttime, but anyway, it releases some enzymes. So background activity presence every time in our digestive tract. It's continuous process. New portion of food increases activity. Hydrochloric acid from chyme are prepared to pass into duodenum due to parietal cells. Then, bile. Bile emulsify fats. What does it mean? Here, protein denaturation. Big molecule, then denaturation, and then breaking down. Fat. But big molecule, big drop, and then it converts to small drops. So emulsification. These drops become smaller, so enzyme has more surface to break lipids down. But bile has more functions. Activate what? I see. Lipase, cholesterol, esterase.
Secretary of the small intestine, yes?
Increase activity of secretory cell intestine helps to release intestinal. What more?
Protective.
And so when the bile enters the intestine, its acid part absorbed with the blood goes to the hepato to release the bile and
It helps bile components helps to increase bile production. We have two processes: bile production and bile release. Bile production occurs continuously. Bile production, liver. Duct, liver duct, bile production, synthesis, no? Bile synthesis or bile production. It occurs continuously. Speed of bile production increases after meal, of course, but it happens continuously. Production between meals, bile is collected in bile bladder, inside bile bladder, And here, bile gland can control release of bile in response to food process, by portion, step by step. Значит, bile production, bile release. Here, bile release. What hormones affect mostly bile production? Secretin increase bile production. So cholecystokinin increase
Contraction of bile,
Sphincter of Oddi.
relaxes sphincter Oddi, and it increases bile release together with pancreatic juice. So bile helps to release pancreatic juice due to cholecystokinin effect. Значит, conclusion about cholecystokinin and secretin. These hormones are main to provide digestion in the duodenum. Cholecystokinin is released in response to fat and protein products. Secretin to hydrochloric acid. Cholecystokinin effect. It stimulates enzyme release, pancreatic enzyme release, and bile release. Secretin stimulates hydrocarbonates release and bile production. And bile components, bile salts. Bile salts absorbed in the intestine, small intestine, large intestine, because small and large intestine, there is part of small large intestine, works together. They both contain microbiota. Microbiota converts primary bile cells into secondary bile cells, and after each bile cells absorbed by 90%, 90% absorbs into blood, and come back to liver. And stimulates bile production. This bile production is stimulated mostly bile salts from the blood and secretin. Secretin, of course, across blood also, affect across blood, with blood. Bile release is stimulated by cholecystokinin. These two hormones work together. They are not single hormones, and the intestine contain a lot of endocrine cells with different hormones. For example, gastric inhibiting peptide. Gastric inhibiting peptide. I don't have space. Gastric inhibiting peptide.
You don't want this skin from us, right? You ask questions from it, but you don't want us to draw it, right?
No, now I explain everything.
I know that like it's hard on exam to draw
Yes,
anyways.
of course, I don't ask you to draw it. So now I explain step by step. I want to look at your presentation. About I didn't draw it. Okay, gastric. I will not explain about gastric inhibiting peptide because it's so much for dentistry student. So just for medicine student. Well, now about yes, insulin. Very important to understand the connection between metabolism and digestion. In the key point, insulin production. What factors stimulate insulin production?
The amount of
Glucose where?
in blood.
Glucose of the blood, the amount of glucose, level of glucose in the blood, increase or decrease glucose? Increase glucose concentration in the blood.
And glucagon.
Значит, glucose stimulates production and release insulin and inhibits glucagon release. Opposite effect. What happens with glucose during meal? During eating, what happens with glucose?
Increase
Increase due to storage. Storage release glucose. Glycogen releases glucose from storage at first, during a meal, immediately, due to reflexes. And insulin concentration start to rise during meal, before intestinal phase, so during cephalic phase. Then gastrin from here also stimulates insulin, gastrin, and cholecystokin. So during gastric phase, more insulin. During intestine phase, more insulin. There's a lot of factors, and vagus also. There's nervous regulation, humoral regulation during each phase, cephalic, gastric, and intestine. Beta cells produce more and more insulin. This is controlled by glucagon. Glucagon has a lot of influences. I will not explain now. It's a very complicated interaction between different hormones. It's so much for you. But it's enough this. But
Obviously, we have a positive feedback, yes.
feedback.
Yes.
Feedback we have, of course, for endocrine cell and endocrine. I will explain how. But it's interesting fact. Insulin stimulates enzyme production. There's another hormone, insulin. This cholecystokin and secretin are main to stimulate pancreatic juice release. Another hormone, insulin, it helps to increase release of pancreatic juice. Insulin. Glucagon has opposite. So what part of our body influence on pancreatic juice release? At first, duodenum factor inside chyme, and secondly, insulin, and endocrine factor. And actually, blood composition, for example, duodenum and blood in connection with pancreas have feedback. What does it mean? For example, pancreas released a lot of enzyme, excess. Some enzyme doesn't have substrate. Excess can be connected with substrate. Trypsin doesn't connect with protein because not enough protein. What happens in this situation? Excess of enzyme inhibits cholecystokin. If you take tub or pillow with the pancreatic enzymes, Festal, Methylene forte, with excess, without receipt, without recommendation of your doctor, it may inhibits cholecystokin and release and inhibit work of your pancreas. It's necessary to take this pill just if you have problem with digestion in the duodenum, problem with gallbladder, for example, with bile release, or with pancreas, pancreatitis, for example, but not a normal condition. Well, next, small intestine. Small intestine, there is contact digestion prevail. Contact or membrane digestion. Contact or membrane digestion. What does it mean? we have two types of digestion: distant, inside lumen, and contact with a very closest contact of food with enterocytes. Enterocytes. Did you study enterocytes in histology?
No,
Why?
we don't have it until now. We study it only in pathoanatomy, just a little bit.
Microbiome.
Yes, yes.
Why in the intestine, small intestine, has a lot of microvilli, big villi for what?
Increase the surface for
Increase surface for the final digestion, including membrane digestion and absorption. With final digestive processes. Enterocytes enterocyte is microvilli. Enterocyte is polar cell. What is a membrane with microvilli? Apical and basal?
Apical.
And basal. Different with different functions. Villi has a lot of enterocytes like this, and special cells, stem cells, stem cells, endocrine cells, inside down, here on the bottom. How an enterocyte works? Regular enterocyte. Surface of the membrane has a lot of glycopeptides, polysaccharides. Glycopeptides has enzymes sitting here. Enzymes. They provide the first stage of the membrane digestion, digestion includes, at first, preparation to digestion, then breaking down to big molecules like here, protein, big molecules, then breaking down to smaller molecules. Finally, mostly monomers. Breaking down to oligomers happens in the liver. Stomach, then smaller, but anyway, oligomers in duodenum. There's maybe some monomers or dimers in the duodenum, but not mostly oligomers, smaller. And dimers, monomers are produced during membrane digestion. Glycocalyx form mostly dimers here, before oligomers, and monomers on the membrane inside. Here, monomers, and the last enzymes embedded inside membrane are located near, closer to transport. It's very important to understand. For example, you mark glucose by radioactive element, glucose molecule, and insert, put it into small intestine directly. Solution with glucose. One case. Second case, you mark glucose in food and then observe speed of absorption from digestive food. Yeah? Broken food molecule. What speed is higher? From glucose solution inserted directly or from digested food? What speed? Speed absorption speed is higher. Higher. Glucose solution or glucose from food.
Solution.
Solution has solution. You mean this one?
Solution has salt glucose.
Yes.
Yeah? What speed of higher? But here we have, for example, dextrine or disaccharide. Disaccharide. And it must be digested and absorbed. And second case, you have solution glucose, free glucose. The saccharide contains radioactive glucose, maltose, marked with radioactive particles, and you can observe it with record. Radioactive radiation. What speed is higher? Absorption glucose, free glucose, or after final?
Free glucose.
After final.
Here, absorption will be faster because the digestion on the membrane. After digestion of the membrane, glucose connects with transporter faster. This molecule of glucose must find its transporter. It requires time. So normal digestion after meal is more adequate. The better assimilation of food is better with natural digestion. Well, mechanisms. Mechanism of absorption. Scheme of mechanism, copy with me because it mechanism of absorption. We can do that. We will study the same mechanism again in the kidney.
Professor, if it is very big, can you write in this?
Here,
Yes, because
Let's have a photo.
atayım işte eskiden.
enterocell. Again. Basal membrane contains sodium potassium pump. It's necessary to provide primary and secondary active transport. Sodium potassium pump. With ATP hydrolysis, active transport. It creates concentration gradient for sodium. So sodium can pass across apical membrane passively, or apical membrane uses concentration gradient, sodium concentration gradient, to pass together something more. For example, glucose molecule or amino acid. Sodium amino acid. But actually, just 20% of amino acid is transported in free form in monomers. More 80% is the protein products is absorbed across apical membrane in dipeptide form, peptide, D or free peptides. With another secondary active transport, I will develop too much. Peptides, dipeptides. And then peptides transform to amino acid inside enterocyte. It's intracellular digestion. Final intracellular digestion. 80%. And then amino acid go in the blood. Intercellular fluid and then in the blood. Glucose also. Glucose. With another transporter facilitated diffusion. Passive transport with special carrier facilitated diffusion. So absorption of glucose, amino acid, peptides require pump work. So this is secondary active transport to pass across apical membrane. Then it's not important. If it requires energy, anyway, it's called active transport. And independently on membrane, apical or basal. Active transport, but secondary active. Because it uses primary active transport pump, sodium potassium pump. What about fat? Fat. Triglyceride. Fat molecules are concentrated together, different fat lipids molecules, triglyceride, cholesterol, phospholipids, are collected together in muscles. Did you study muscles in biochemistry? Biochemistry, muscles. Muscles are big molecules. Micelle contain bile, triglyceride, cholesterol, phospholipids, lipase. It digests lipids inside, and micelle bile salts helps to absorb lipids. Lipids may be absorbed with simple diffusion across lipid membrane, because membrane is lipid, or with special care by facilitating lipid absorption. From this micelle, big molecules, monoglyceride and free fatty acid. With triglyceride are broken down to finally to monoglyceride and free fatty acid. Sometimes maybe glycerol, but mostly this. Then phospholipid can be absorbed. So cholesterol. Without the things, now cholesterol storage is not important. Important is then inside enterocyte, free fatty acid, monoglyceride synthesize again, what? Triglycerides. It's resynthesis. And together with again phospholipids, cholesterol, some free fatty acid, and protein. Did you study other proteins? No? When you have biochemistry exam, next semester. Biochemistry. This semester.
Not very good.
نعم بس أكثر كثير.
Proteins.
مبسوطة.
Other proteins. Other proteins.
Proteins for transport lipids, yes?
To transport lipids. Because lipids are fat soluble. Is not well transported by fluids. It's necessary to have transported proteins. What is it? Other proteins. What is the name of this big molecule, big particle? Inside enterocyte. What is the biggest drop? Kilomicrons. Kilomicrons.
With protein and
Other proteins are transported and different kinds of lipids: triglyceride, free fatty acid, cholesterol, phospholipid, everything. And a lot of triglyceride, a lot. It absorbed into lymph, not blood, lymph. It transported by lymph into liver. Liver. What happens in liver? It converts. Yes. Lipoproteins.
Mhm.
HDL, low, high density, low density, very low density, and we have
HDL or FDL.
between very low density and low density. What is elevate? For physiology, такие medium size molecules, bad molecules having a lot of cholesterol.
FDL is bad for our body, HDL is good for our body.
Yes, because HDL contains less cholesterol, less triglycerides. Lipoproteins like low density, very low density contain a lot of cholesterol. Well, it's not absorption, motor function.
Lipoprotein portraits.
Yes, portal vein into liver and then into blood circulation. After liver, at first portal vein to liver, portal system, and then transformation to different lipoproteins, depends on the situation, and then into blood circulation to transport to every cell, blood vessels, and exchange with blood vessels. Blood vessels and cells by cholesterol, phospholipid, free fatty acid. Very motor function. Motor function. Again, stomach. After swelling, and during swelling, process swelling. What happens with stomach motor activity?
It's increased.
Increase or decrease?
Increase.
During swelling.
During
What happens when you do swelling,
And it decrease.
increase, decrease, relaxation, fundus relaxation, fundus relaxation. Actually, fundus has a low motor activity. Tonic activity prevails. What does it mean, tonic activity? Tonic activity means waves of membrane potential without spikes, and without strong motor contraction. Invisible activity. You can't see it. You could watch animation in my lecture, in my video lecture. Animation what happens in stomach between meals and during meals. Between meal just tonic activity. When you are eating and swelling, at first the relaxation of fundus to increase volume, because stomach has a storage function, food storage, to collect more and more and more. Storage. To increase volume of stomach, to make more stretching. Then, during eating, pacemakers centers, motor centers, smooth muscles, pacemakers, located in the beginning of body, stomach body. It creates waves of peristalsis. Peristalsis. Where peristalsis is more, in the body or in the antrum? Antral. Division responsible for motor function, motor activity. Fundus has more secretory function. A lot of cells, parietal cells, chief cells secreting gastric juice. Antrum doesn't contain parietal cells, absolutely, and the little chief cells, because it's responsible for motor contraction. At first, slow digestion due to secretion of gastric juice, digestion of pepsin, by pepsin, proteins by pepsin, then movement of chyme into antrum, pyloric sphincter is closed. Strong activity, motor activity in antrum, providing grinding, mixing with gastric juice, like mixer. And when chyme becomes acidic and has smallest particles, it opens pyloric sphincter and some food, not every food, and not all chyme, some portion goes to jejunum and continue digestion. You must know exactly names of motor contraction inside intestine. What are the names? Tonic, background, peristalsis, segmentation, rhythmic segmentation. But three types, main three types. Tonic activity doesn't have visible contraction. It's fluctuation, waves of membrane potential. It's prepared background, to be prepared to signal. If I can draw, for example, membrane potential, smooth muscles during tonic contraction, membrane potential. Resting membrane potential, then slow waves. slow waves. It doesn't reach threshold, doesn't have spikes, and no visible shortening. You cannot see something intestine. But after stimulator, vagus nerve, local nerves, gastrin, motilin, or hormones, acetylcholine, for example, acetylcholine increases waves, it reaches threshold and produces spikes, action potential. Action potentials triggers smooth muscle contraction, shortening. You must draw peristaltic reflex R. It's present in presentation. My presentation I explained in previous class. And last about hunger and satiety. Hunger and satiety. Yes, yes.
It's more understandable than this one.
Local reflexes providing peristalsis prevails in the intestine. Small intestine, large intestine, mucose, layer of circular muscles, layer of longitudinal muscles. Two plexus, submucous plexus, nerves, intramural gut, submucous plexus, and myenteric. Myenteric plexus. It contains neuron. Both of them can contain afferent neuron, but afferent neuron are mostly located in the submucous plexus. Afferent neuron higher. So I will draw in accordance with this rule. But actually, all that neuron may be located in both. Afferent neuron, Doggett diet. Did you study Doggett's? Doggett? Forget. Physiology doesn't require, but maybe histology requires. Maybe in medical institute. Afferent neuron. This is a special type of neurons. These neurons have several active fibers. As you know that unipolar neuron, pseudo-unipolar neuron, bipolar neuron has two fibers, afferent neuron. Dendrite and axon. But this neuron can have several receptive endings. Like this. A receptor and one axon and another axon. Maybe so. Afferent cell directly or interneuron, and then afferent. There's maybe different variants, not common scheme. Afferent neuron, interneuron, afferent neuron, or afferent neuron indirectly afferent. Monosynaptic, polysynaptic. This reflex doesn't have a nerve center inside central nervous system. It has local nerve center, So we can say again afferent and afferent in accordance with intramural centers, intramural neural network. What is the idea to form local reflexes? Due to divergence, there's one afferent neuron, and its axon can be divided to two parts or several parts due to divergence. Do you remember divergence?
Mm-hmm.
One axon divided divergence and form two neural chains. Afferent neuron 1 afferent neuron 2. For example, afferent neuron 2 is inhibitory, afferent neuron 1 stimulatory, excitatory, inhibitory. So it inhibits circular muscle. This part, second part, and next part, stimulation. So we have large diameter before, and then contraction, like wave. And you can draw different principles. What about longitudinal? Reciprocal. Another branch, inhibitory afferent neuron and inhibition. It's contraction of circular muscle, inhibition of longitudinal. The same recombination, just principle. There are a lot of combinations, and you must understand just principle. What about Hunger satiety? You must know at first the main centers located in hypothalamus.
Lateral and MVH.
It's old knowledge. Now science knows a little more about the hypothalamus nucleus. For us, enough this. Destruction of lateral hypothalamus and animal doesn't eject food. But if you maintain its homeostasis, with time, with several days, this animal starts to eat less than before, but starts. It means that not just hypothalamus control hunger behavior, eating behavior, but hypothalamus is most if you damage ventromedial hypothalamus, an animal starts to eat so much it can stop it at the next time. But with time, this eating behavior reduces and becomes stable. And two principal phases. Hunger satiety, sensory phase and metabolic. Sensory hunger is directed to prevent exhaustion of storage. To have storage. The first hunger you feel approximately in 4 hours after meal, when you hide inside duodenum and digest and absorb. But empty stomach sends signal, I am empty, I need food, to continue digestive process. Then body starts to collect storage to decrease nutrients in the blood to increase your hunger, stimulate hunger cycle. If you don't eat, then second phase, metabolic hunger, when body uses storage and cells become less active. Satiety. What is the function of satiety? To stop eating. The first stage is started to signals from oral cavities in the stomach, then increase nutrients in the blood, glucose release reflex, and then information from hormones, cholecystokinin, insulin effect on brain, and then during time 2 to 3 hours, another hormones, complicated hormones, peptide YY, and other hormones prolong satiety. Well, see you during grazing time and